All right, so we're going to welcome everybody to the first of our habitability seminars for this semester, and just kind of a couple of upcoming things. And I'm going to let Eric Hiatt, who's Andy's official host, do the introduction. That we now have a full calendar of speakers, so we'll get the calendar up, but we'll continue with the standard second and fourth Mondays for the seminar, and we'll continue with the tradition of the third being our general meeting. In addition, we have our retreat coming up for those who are registered, and there's probably a couple spots left if you still want to come to the retreat on February 16th. Brandon.
One quick correction to that. In April, because the seminar would have been the week of the eclipse, we're not having a seminar there, but we are having one the last Monday of that month. So there's still two seminars that month.
Right. So we'll put one on the 5th Monday of April.
The eclipse.
Right. Yeah. And the eclipse will not be any activities here. Okay. So I think with that, we'll let you take over.
Awesome. Thank you. Well, it's my pleasure to introduce Andy Czaja. Andy's from my hometown, Cincinnati. He was gracious enough to let me talk in front of his group, and we reciprocate and try to bring him down here. He does a lot of interesting work that's Mars related to me. But Andy started out with his BS at the University of Connecticut in environmental science and biology, and went on to UCLA for geology and paleobiology. He was followed up with a postdoc, I believe, in Wisconsin.
Yes.
And now is an associate professor at the University of Cincinnati. He's also a member of the Mars 2020 Science Mission, the Return Sample Mission, and a Campaign Science Group member as well. And so with all due, Andy's going to give us his talk on Earth's ancient biosphere.
Thanks. Thanks, Eric. Yeah, I really appreciate the opportunity to come and talk to you all. Yeah, so I'm interested in setting up, potentially down the line, I'm thinking long-term, a center at the University of Cincinnati, but we don't have as many people who are already doing things like planetary science and integrative biology and things and physics to bring together. But I'm sort of thinking maybe down the line we can have justification for hiring more people who are doing these sorts of things. Because right now it's largely me doing planetary science. And there's some people, there's at least one faculty in biology who's doing some astrobiology relevant things. So I'm really happy to get to be here and to meet a lot of you and learn more about your center and just see the school in general. Yeah, so I'm going to talk about, as Eric said, the Earth's ancient biosphere, because that's what I directly study. I study ancient life on Earth. I'll say more about that in a moment. And I use it as an analog for life in the universe. And as Eric mentioned, I'm also now a member of the Mars 2020 mission, have been for the last 4.5 years or so, actively, exploring Mars. Prior to that, it was more theoretical, like, oh, this would be cool if we could, explore other planets to look for evidence of life. And now I actually get to do it, which is pretty cool. And I'll say more about the Mars. Okay, so there's a little bit more about me. Again, my main research interests are the search for and study of ancient biosignatures, and that largely is microfossils, fossils of ancient bacteria, archaea, microbes, whatever they were at the time. Mostly more than 2.5 billion years ago, so into the Archean time period, using things like co-located microscopic optical and chemical imaging with Raman spectroscopy and confocal laser scanning microscopy. That's what I can do in my lab. I also have done some isotope work, which I have to go out and do that with other folks. And I'm interested in kind of increasing this capability at UC to explore for other biosignatures or explore these microfossils in other ways, with an eye toward being able to do that for samples brought back from Mars someday. And again, yeah, the search for past or present extraterrestrial life, especially on Mars, is another interest, of course. We don't have any of those samples yet to do that with. We can explore with the Mars rover, at least somewhat. Okay, so I like to put this up near the beginning just because I always run out of time in my talks. And so I just want to thank all of these folks for help over the, currently and in the past for different aspects of what I'm going to talk about today. including at the top there, 1, 2, 3, 4, 5. I don't know why there's a tab there before Kira's name, are all current and or former students of mine who contributed to this work. And of course, the funding that makes it so possible. And we'll see some direct things from the 3 of my students on that list, and you'll see them in a few minutes. Oh, I should also say, I like to keep my talks a little bit loose too. So if you have any questions, please raise your hand. You know, if I don't see you, call out and I'll answer questions as we go. I don't want to, you sit there and wait until the end to ask me, like, what did you mean by that? And it meant you didn't understand anything else I said. So I'll try to explain. I know this is an interdisciplinary group. I like being in interdisciplinary groups, but I think most people were, guilty of talking in jargon sometimes. So this is a real general overview of today's talk. Talk about life on the early Earth. I had more things here, but I decided to simplify it. We're talking about some of the things that I do toward that, and then, how this applies to searching for life on Mars and how we are doing that with Mars 2020. Okay, so getting to life on the early Earth, there is several types of evidence that we can look for. And they can be broadly grouped, I would say, into these 2 categories, morphological or geochemical. Right? Morphological are portions of the actual organisms that lived then, or some object that they made, like what we call microbialites, including, you probably heard the term stromatolite before. This is a microbial feature, right? It's a fossilized microbial mat. So it's not the microbes themselves, but it's evidence that they were there. So those are 2 of the main types of morphological evidence from the early Earth. Of course, you get in, when I say early, again, I'm talking Precambrian, I'm talking Archean, 2.5 billion years ago. There's no plants, no animals, no fungi, none of those ones with bones and hard parts and things like that, or complex structures. And a more common type of evidence is geochemical, because these fossils left behind portions of themselves, which are too small to see or to be identified as being part of the object. But we can say, by detecting them, say, life must have been here, life leaving behind this signature, like organic biomarkers. So organic molecules that are specific to life. In some cases, they are not very specific, but it's like, this is too complex to have just formed naturally. In other cases, you can try to get toward larger taxa. We're not identifying species with that. This is not what I do. I'm not an organic geochemist, at least not for detecting organic biomarkers, but that's one thing that a lot of people do. We can use isotopic proxies. So different organisms will fractionate different elements to different degrees. They prefer either the lighter or the heavier. It's usually the lighter isotope often. And so you can detect different, types of these elements that we call isotopes in the rock record, which can tell us, or at least help give us confidence or some evidence that life was there. Certain times elemental abundances can also be either formed by life or by some process that life did. And then sometimes specifically trace elemental distributions as opposed to just the larger elemental abundances. But I'm not going to talk about all this today. This is just kind of an overview. But I mention a lot of these things too, because if you find evidence of life on the early Earth, you often need to find a whole bunch of pieces of evidence. So it's not enough to say, I found this one object and it looks like a fossil bacterium. If you found that in rocks that were, 700 million years old, or even maybe 1 billion years old, people would probably be like, okay, because we have pretty good confidence that life existed at that point. It wouldn't be that interesting to find one. But if you found one object in a rock that was 3.5 billion years old and tried to write a paper about it, you would never get it published, because it's one thing. As we'll see in a moment, the morphology of microbes, actually right here, they're typically coccoids and filaments, balls and sticks. That's what these things look like. Sometimes they're a little bit wavy. It's not that specific of a morphology. And especially when they've been sitting in a rock and degrading for billions of years, right? They don't look exactly like they did. Just like you find a dinosaur bone, that's not what the organism looked like. That's what part of its inside looked like, but it's still also altered. It's often deformed and, you know, stained and things like that. Same thing with fossil microorganisms. They get altered from what they looked like originally. You know, if you see pond scum on a body of water here, you can use your naked eye to see, oh, there must be microbes there. There's algae, there might be cyanobacteria. You got a microscope, you can tell the difference between them. But you know they're there because you see this green scum, and sometimes you see bubbles, and you can tell there was like some sugary material in there that they use to stick themselves together. Once that stuff gets fossilized, that green color goes away, a lot of the chemistry goes away. You know, you need more information than just seeing them morphologically, unfortunately. That's the first thing, because if you are looking for fossils, you need to see them first before you're going to do more work on them. And then the more work you might do is to try to characterize that morphology more clearly by looking at to see, do these things have a cell wall? Do they appear to be in a biological size range? And then you want to look at their chemistry. Is it made of organic material, or is it made of some mineral? Maybe you can study the carbon isotopes of that organic material. You want to find a bunch of specimens. Again, one is not going to convince anyone. 2 or 3 probably won't either. Because if one thing can get preserved, a bunch of them should be able to get preserved. You might be able to find multiple taxa, because most organisms don't live in a monoculture on Earth. They live with lots of different types of other organisms. Oftentimes similar, ones that have similar environmental needs. And you want to know that you want to find them in a biologically plausible environment, right? If you find them inside preserved somehow inside a vesicle in a basalt, if you did, you'd say, well, maybe that must have colonized later, right? Because it's not going to live in that thing when the basalt was when that was magma or was lava still, right? You want to find these in relatively cool, marine environments, maybe lacustrine environments. And their preservation should match what you know about the rock, right? The geologists in the room know when you look at a rock and you can tell how it's been altered over time since it formed, if it has been. Well, the fossils inside should match that, right? If you find things that look really well preserved inside a gneiss, it probably was not originally in that rock, right? that gneiss has been, hugely heated and pressurized and deformed. You shouldn't find fossils there. So anyway, this isn't to say you have to step through and find evidence of every single one of these things before you can say, hey, I think I found life. The more evidence you have, the better. So with that, here's what we know. This is a geologic timescale going from the present over here at 0 billion years ago to a little more than 4.5 billion years ago on the left side at the formation of the Earth. This is from a textbook, an intro textbook, so it's a little bit simplified, but it shows all of Earth history here. And what do we see in terms of evidence of life? There's a lot of geological things early on, but we have some evidence from isotopes, from geochemistry, at about 3.8, 3.7 billion years ago, that there may be some evidence a little earlier than that too, that there was life, carbon isotopes and iron isotopes too. I added the iron isotopes because that's what I studied as a postdoc and just didn't make it to this textbook, I guess. Well, this textbook came out around the time I'm not the only one who did it. Other people were doing that work too, but it was relatively new at the time when this textbook was published. But maybe more convincingly, in terms of morphological evidence, we have stromatolites, again, those fossil microbial mats. We have microfossils that date back to 3.5 billion years ago. Some are better preserved than others. Some are more convincing than others. There's a lot of debate in the literature about whether some of these things are actually evidence of life. But I would say the consensus is that, yes, we have at least stromatolites that go back to 3.5 billion years ago. And so that's the time period we're interested in, and a little younger than that. And the reason it doesn't go back further than that is there's 2 reasons. One is these microbes are soft and squishy, or were soft and squishy when they were alive. They don't preserve very easily, right? They didn't have bones. They didn't have teeth and hard parts. That even those, like, you know, we're not finding dinosaur bones all over the place. They lived all over the place, but most of them, you know, didn't get preserved. Part of that is because they lived on land. Land is not a great place to preserve things because that's an erosional environment. The microbes living in the ocean, that's a depositional environment, but they're soft and squishy. They don't preserve easily. The other thing is plate tectonics, or just general, you know, alteration of the surface of the Earth. And this is a figure from a book. It's not even a textbook, but it's a book about the search for early life on Earth. And this is my advisor, Bill Schaupp, wrote this book. And he has this quote here: the older the rock, the greater the chance that it no longer exists. All right, we've been forming rock all throughout the history of Earth. But if you look at the ages of the rocks that exist today, rocks that are, you know, about 4 billion years old or 3.8 or so are very, very few. Rocks that formed recently, there's a lot more of them. And this is because we're continually recycling the Earth. Keep this in mind when we talk about Mars in a little bit, because Mars has had a very different history. Okay, so now I'm just going to talk a little bit, give a little real brief view of some of the actual fossils of the ancient fossil record and some of the work I've been doing. And I should really keep an eye on the time, but there's this clock here. Okay, I think I'm doing okay. Perfect. Thank you. So again, the fossil record that I typically study is morphological evidence, microfossils and stromatolites. Stromatolites look like things like this. You can maybe make out. I apologize for those in the back. It's, you know, it didn't come out as great, but there's some little wavy lines on there. There's these sort of branching structures here on this middle kind of orange panel. Here, let me point with my mouse for those of you online. So little wavy structures here. There's kind of these columns that are branching through here. These are called stromatolites. This maybe we call more just a microbialite. It's more flat lying, but occasionally there's these little peaked structures. These are all cross-sectional views down through them. So we're looking at an accumulation of these sediments over time. And then we have some fossils over here on the right side of the screen, microfossils. And these are all particularly well-preserved examples. There's some that are much less well-preserved. But, you know, we've got tubes, some bendy tubes, and we've got some you know, spheres. That's largely what we had. There's a couple other examples I'll show you in a moment. This is just to remind us what it is we're looking at. So if we look at the Archean, this is the time period from 2.5 billion years ago until, it actually goes back a little more than 3.5. It's more like closer to 4 billion years ago, maybe 3.8 billion years ago. People are actually still working on exactly when the boundaries are of these time periods. Notice here, I listed it in millions of years ago rather than billions. So it's 2,500 to 3,500 million years ago. These are all the occurrences, and I think I need to update this. This might be slightly out of date, but anyway, there's not that many more known. These are all the occurrences of microfossils in that time period, right? And it's because there's not many rocks in that time period. It's not because we don't find many. It's also because we don't find a lot of fossils in the rocks that exist, but it's also because there's not many rocks that are there compared to, you know, more recent times. But there's a bunch of different morphologies. I mean, there's rod-shaped ones, which kind of look like E. coli, that sort of morphology. We've got lenticular bodies, these ones up here that are kind of weird with, you know, these pointy structures on the end. We've got small coccoids, we've got large coccoids, we've got extra large coccoids. There's a scale bar here of 50 microns. This thing's about 300 microns across. We've got narrow filaments, we've got broad filaments, and then broad tubular filaments. Because sometimes the filaments of cell, it's a chain of cells in what we call a filament. Sometimes you find the actual chain of cells. Sometimes you find the sheath that they existed inside, because some microbes excrete a sheath around themselves that they use for protection and actually motility. And a lot of times the sheath gets preserved and the cells do not.
Excuse me?
Yes.
When you have those chains of cells, is that an actual multicellular?
Not really. It's more of a colony, because you can separate them into individuals and they would continue to reproduce and form a new chain. Maybe it's on the way toward multicellularity, but this is long before we have any evidence of true multicellularity. Like a multicellular organism like you or me, if we took ourselves apart into component cells, that'd be it, you know? Whereas these things, yeah, they're all, I mean, it's a little more complicated than that. One sec. With certain cyanobacteria, they do sometimes have certain cells that are specialized, and they do nitrogen fixation. But even so, it's still not true multicellularity, but it's maybe a blurry line. Yeah.
Can I ask a naive astronomer question?
Please.
So these are all really small.
Yes.
And they're found in very old rocks.
Yes.
So how does the field work actually work to go and find these? You go somewhere with very old rocks.
Yes.
What do you do?
Well, so excellent question. No, that is a very good question, and one I should have probably anticipated and talked about. But so one of the things you do is look for things like this that you can see. You see there's a hammer here for scale. This one, there's a pen right there for scale. Again, I apologize to those of you online. I'm pointing at the screen instead of using my cursor. In fact, let me add a laser pointer. So there's a little pen right there, and there's a hammer over here. These stromatolites, these microbialites, you can see when you go out into the field. So that's kind of your first clue, like, hey, this is an environment that is potentially, that was habitable, that things did inhabit. So you might then take a sample of this stromatolite. It turns out, though, a lot of times in these stromatolites, you don't actually find fossils of the organisms themselves. And that's often true, especially of the ones that are composed of the mineral carbonate, either whether it's dolomite or calcite. So those are two different carbonate minerals. It doesn't really matter. The way the mineralogy, the way that rock changes over time, it destroys any microstructures in there, structures at the scale of these microfossils. You can still see the layers in the rock. Sometimes they are silicified, meaning it gets either replaced by silica, or in some cases, silica may be the mineral that precipitated originally. Silica preserves fossils really well. If you get these microcrystalline silica, it actually takes a lot more alteration, a lot more heat to cause those minerals to change and grow, and the fossils are better preserved. All of these are preserved inside silica. These fossils here are still trapped inside rock. The way we find them is we go to the field, we collect samples of, we search for chert. Chert is a rock that is made up of microcrystalline silica. And you want to look for chert that is black or dark brown, because that means there's organic matter in it. But you bring that back to the lab, you make what we call a thin section of it, a very thin slice of rock that's on the order of about 100 microns or thinner. And it's then transparent. You can put it under a microscope and you can see if there's anything in there, if there's any fossils in there. So we have no idea when we're in the field if we found anything. Aaron can back me up on that. He and I did some field work together a bunch of years ago now. Boy, it was almost 10 years ago was the last time. And yeah, if you know you have stromatolites, you don't know if you have any fossils. I brought back a whole bunch of rocks that I haven't found anything in yet, but I did bring back some that I did find rock fossils in. This one's a little different. This panel, what's called D, I grabbed that from someone else's paper. That is a sandstone. So a sandstone is a rock made up of, sand-sized grains like beach sand that all got, cemented together. So that's a shallow water thing. Those don't make good thin sections, but that's an actual slab of rock that they just cut and maybe polished a little bit, and you can see those structures. But again, we don't find the actual microorganisms preserved inside there, just the structures. So thank you for that. Okay, so now I'm going to tell you about a few examples that I've found in my own lab. So this is from work I did in South Africa. Here, you see the map here of South Africa, and there's this chunk of rock called the Kapval Craton. A craton is just a big contiguous section of rock from a certain time period. So a lot of time cratons from different times or different places get shoved together, over time to build up new continents as plate tectonics occurs and chunks move around. Anyway, this craton was formed early on in Earth history, and there are rocks preserved here between, well, the ones I looked at were between 2.5 and 3.5-ish billion years ago. There may be rocks that are a little younger than that. I'm not sure there's rocks that are much older than that on there. And the place we're going to look at is over here on the western side of it, up in sort of northwestern South Africa. And here's a geologic, map showing the area. It's large. This little brick pattern indicates it's largely carbonate. And on top of it, there's something called the Kuraman Iron Formation. Which is a time period where a whole bunch of iron was deposited down on top of that because of the ocean chemistry. I'm not going to talk about that right now. And here's a strat column over on the right side showing, this what's called the Campbell-Rand-Malmani carbonate platform. This formed on the a shallow marine area just offshore. So there was to the what's now would be the east of here. I guess there would have been dry land, but then it got deeper as you went toward mostly to the west, but a little bit south. And as this was, this rock was there, the platform was there. There was this marine carbonate being deposited, and we got a lot of stromatolites of different types, indicating different water depths. And overall, this was getting deeper and deeper and deeper. Either the land was subsiding and the water level was getting deeper, or sea level was rising. It's hard to tell the difference actually in the rock record, or it could have been both. And so overall, there's just different the point is there's different paleo environments. There's deep water environments, there's shallow water environments, and we find different types of fossils in these. So here's just that context. Aaron has seen this place before. This is all this gray rock here, sort of grading up into more browner rock. It's a little hard to see the rock at the top. It's a little browner here. Is all carbonate. It's all stromatolites. As you go up, you see these big domal microbialites. Because again, you don't see this happening now. You don't find these sorts of massive deposits of stromatolites now, because we have things like snails and fish that eat microbial mats and mow them down like a lawn, right? They didn't exist back then. It was just microbes. So microbes just, went crazy and made these giant mats. And this hillside over here, that at the very top, those layers you see, that is that Kuraman Iron Formation. So this whole area is capped by iron being deposited, and that occurred in the deepest water that we saw in this area. It looks like a different hill, but you go up over the crest of that, and then it just sort of is flat for a bit, and then it starts going back up again. Okay, so I say newish fossils that I'm going to talk about now. These were actually originally described in 1987, so they're really not that new anymore. But they were interpreted at that time as, it still seems newish to me. I was 11 at the time, so I was around, but I didn't know anything about this, of course. These are up in that real deep water, not in the iron formation itself, but in rocks that had a lot of iron in them, and it's interpreted to be really deep. But they were originally interpreted as cyanobacteria, right? Cyanobacteria are microbes that, in today's world, that they're the oxygen-producing prokaryotes, organisms that don't have a nucleus. We've got single-celled algae that are more closely related to plants that do the same job, but they're a more recent evolutionary stage of life. These things were the cyanobacteria we think existed back this far. That's where the oxygen came from to oxidize all that iron that fell out to make those iron formations. But cyanobacteria don't live in deep water. Why don't they live in deep water? They need sun. They do photosynthesis. They need sunlight. So it's a little hard to reconcile the fact that these seem to have been deposited in deep water, and it doesn't look like they washed into this deep water zone. It looks like they were living there. So the work I'm doing is trying to think about maybe these are something else. Maybe they're not cyanobacteria. Yeah, because it's the fact that they're cyanobacteria is based on their morphology. They're large. They have these tubular sheaths that some cyanobacteria now do too. And they said, well, it does seem deep, but because we think they're cyanobacteria, maybe it wasn't too deep. But it's sort of a circular logic there that is you know, at the time it was like, well, we found these things, so we're publishing it, and this is our best guess, but we don't really know. So I have a student now, Sam Hall, who's doing some work on this. I've had some other students work on this over the years too. But this is, I showed you those images from on the previous slide. These are from the original publication in 1987. And that's, you know, the figures don't look that great. It's hard to see stuff. So we've taken our own images. This image here is of the same piece of rock that they found in that original paper. It's kind of an interesting story about that. We can talk about later if anyone's interested. But I have found them now in other localities of the same geologic unit. So other places where the same layer of rocks exist. And they seem better preserved. And you see this tangled, maybe you can make out kind of this tangled mat. All the little circles you see, that's a tubular fossil kind of sticking out at you. That's cutting cross section. And you see some linear features. That's a tube that's cut this way, cut the long way. Down here is an individual tube that's for whatever reason preserved on its own. Lost my laser pointer, there it is. So that's another thing my student is studying is why are these so differently preserved? Why do they look so different? But I'm going to zoom in on this for a minute so you can kind of see that a little better. You can imagine like a plate of spaghetti, or just taking cooked spaghetti, or maybe bucatini, because that's the one with the hole in the middle, right? Throwing it on a table, and you know, it's just this tangled mess, but everything is largely laying in one direction, right? Because if these things were forming on the ground, on the surface, you wouldn't expect them to be sticking straight up, right? They're going to be mostly this way, but sometimes going up over top of other ones. That's what we see here. And they're also, you can see that sometimes they're squished because these things were soft and they got compacted a little bit, but mostly by the weight of the rest of them. This isn't compaction from the rock during the lithification of the rock, because some of them are not. You see circles there. Those were not flattened. So the ones that are flat probably just collapsed before the whole thing got preserved. Here's just that other example I showed you of this same tube. You see it's about 25-ish microns across in diameter. That's actually pretty big. This is a CLSM image. I apologize, it didn't come out super well on here. Yeah, anyway, I'm going to skip past that because you can barely see it. But what I'm showing you here is what you should be able to see is that there is it's hollow on the inside. That's what you'd expect. If this was a sheath, you'd expect a wall with the inside just full of the silica. If you found a solid tube of organic matter, that's not a very biological morphology, right? Cells are not solid balls of material. They have liquid on the inside. They have a lot of stuff, like a nucleus for eukaryotes, with our DNA and ribosomes and Golgi apparatus and all the other things that these things had. Well, I guess microbes back then didn't have all those things, but those things break down easily. The sheaths survive pretty well, and cell walls survive pretty well. But so you expect a hollow interior, or at least not hollow, but filled in with the mineral matrix. We can also do Raman spectroscopy on these things and say, hey, look, this is made out of what we call kerogen, this dark material. We map it. The green is kerogen. Kerogen is what we call this macromolecular organic material. It doesn't have a specific chemical structure or crystalline structure or anything like that. It's just messy organic material. That's what it turns into during the process of this stuff being preserved. And the red around it is the quartz matrix. There also happens to be a little carbonate mineral up there. It just didn't show up in this particular spectrum. Okay. Yeah, and there's all these preservational differences. I showed you something like that one on the left before, the tangled mass of bucatini. But these two little ones up here, this is notice a different scale. We have a 50 micron scale bar here, which applies to the images on the right, 100 micron scale bar on the left. So these things here are actually cylinders that are degraded. So there's a Raman map. We zoom in on that. It's kerogen. It looks mostly black, but it is a bit greenish. And the red is the quartz again. Raman spectroscopy, I'm sorry, I didn't describe that. That is a technique where you can measure the chemical composition of a material in this case, on a micron scale and map it as well. So you can determine the composition of whatever it is you put underneath it, essentially anything. Now, if we make a CLSM map of this one, I have a little movie here. I'm definitely going slow. Is that going to start? Oh, do I have to turn off the laser pointer? So this is a three-dimensional reconstruction based on fluorescence of these fossils here. And watch as it comes around. And as it comes around right about here, you can see those things are cylinders. But those cylinders are highly degraded, and the view we happen to see through our thin section is slightly to the side. So when I first saw those, I thought, oh, that looks, something about that, it looks interesting. I see like a pattern there, but I couldn't tell. I thought it might be like a spiral, like there are some bacteria that are actually form spirals. And I thought maybe it's that, but when I made this three-dimensional image of it, I could see that's not the case. It's just some little bits of it happened to get preserved and other parts did not. So that's the power of this technique to study these things on a micron scale. So another possibility for these things is that they are, you know, one interpretation, again, the original one was that they were some sort of cyanobacteria, like these modern oscillatory which form these chains of cells that have a sheath around them. You can't see the sheath very well in here because the cells are still there, but the sheath is basically just a clear tube. It's like a straw, and the cells reside inside there. And you can tell that's alive because, you know, it's green, right? We don't get that information in the fossils. There are things called, there's sulfur oxidizing bacteria, and there's a particular taxa called Beggiatoa that, for all the world, looks an awful lot like these cyanobacteria, except the color. In the modern world, you can easily tell the difference. If you just take the sheaths and you degrade them in a rock for 2.5 billion years, it'd be hard to tell the difference. So we're working on ways to maybe try to more confidently to assign it to that and not just say it could be. They do often have sulfur inside their cells, but that it's hard to preserve that too. We're looking at ways to see if we can detect any of that sulfur, if there's any chemical evidence that might get preserved. Okay, I'm going to skip past these for the sake of time. Some other possible sulfur oxidizing bacteria. This is some work that related to what my student Andrea is doing in South Africa on some of these stromatolites. But again, just some of the large coccoidal type fossils. You saw some images of these before. Okay, so what else are we doing with this? Well, we're currently searching for, and in some cases finding, more fossils, trying to increase what we know about the ecological diversity at this time. It's challenging because there's not a lot of fossils that get preserved in any one place. So it's hard to reconstruct an ecosystem. There's also certain types of bacteria that just don't preserve very well. These cyanobacteria, or ones with thick cell walls, or these sheaths, preserve well. But things like sulfur-reducing bacteria, iron-reducing bacteria, they're much simpler. They've got, I shouldn't say simpler, morphologically, they're a little thinner. Their cell walls, they just don't survive well. Things like E. coli, you're not gonna find those in the rock record most likely. But we can also potentially study metabolisms with isotopic analyses by studying the individual fossils for carbon isotopes, maybe sulfur isotopes. And we want to use this to try to help interpret the biosphere prior to what's called the Great Oxygenation Event, which occurred right here. So this chart you see is, no, it's right there, from paper by a guy named Tim Lyons et al. In Nature. We have time on the horizontal axis, again, now in billions of years, and we have atmospheric oxygen content on the vertical axis. The Archean, prior to 2.5 billion years ago, maybe 2.4 billion years ago, there was essentially no oxygen in our atmosphere. But there is evidence that oxygen was produced at that time. It just got scrubbed out of the atmosphere by other things. And then it increased at this time, around 2.4 billion years ago. We don't fully know the reasons for it. I also don't have time to go into all that detail. It's not what this talk's about. But one thing I'm interested in is, yeah, when exactly did oxygenic photosynthesis start? Can we find evidence for it? Why did it, if it started, 3.2, 3.5 billion years ago, like some people have suggested, why did it take so long to for the oxygen to build up in the atmosphere? There is evidence that it goes back to 3.2. The evidence for 3.5 is, I don't think that's very good, but we're still working on that. In fact, I have these little blue lines, the blue arrows indicate what they're called whiffs of oxygen. That was a term coined by a guy named Ariel Anbar in 2007, where there was evidence for little bits of oxygen in different systems, but it wasn't enough to fill up the atmosphere or fill up the hydrosphere. It just was produced locally and consumed locally. So I have another student, Brianna Oriel, and she's working on a project studying and looking for more evidence of ancient oxidation in the Moody's group in South Africa with some drill cores that were collected recently, scientific drill cores. So this is a map showing a couple of places. It's not really important to look at the details on here, but this is a marine deltaic system, so near shore, lots of sand being deposited and microbes growing on that sand. And anyway, that's that image I showed you before of the sandstone with the little peaked structures came from this Moody's group here. And so she's looking at some of these sandstones. Here's a cross section on the left is a thin section of cut cross sectionally down through some of these layers, and you can see some of those darker layers. I have a colleague down the road here at Texas A&M. I know it's not literally down the road, but it's here in Texas, who does X-ray fluorescence mapping. So the different colors represent different elements. And so we can look to see where you see yellow dots, those are pyrites in the rock. And where you see some of these other colors, you've got chromites, you've got clays and things like that. So we can tell what's in there. And my student is doing Raman spectroscopy on here and looking at the mineralogy. Again, Raman gives you it gives you composition, and you can identify minerals. So she's identified, well, it's almost all quartz, but there's some rutile, magnesiochromite, muscovites. There's also, I'll show you pyrite in a moment, but these are all various different minerals that you might expect in these types of environments. The rutiles, chromites, pyrites are these heavy elements that are interesting. This is a pyrite, both in reflected light on the thin section and in transmitted light. Pyrites are opaque, you can't shine light through them. But the chemical composition of pyrite is iron and sulfur. And that iron is in what we call a reduced state, meaning it's not oxidized. If there was lots of oxygen around, you wouldn't expect pyrites to exist, right? Because they go away. But it turns out there's kinetic barriers to that happening immediately. So we might expect some pyrite. Oh, I should point out the fact that this is round or rounded means this pyrite tumbled along a stream or tumbled along a shore or a stream to get to the shoreline, just like a sand grain, and got rounded off. There was lots of oxygen in the atmosphere. You wouldn't expect it to survive. It would turn to rust on the way. But we do see evidence with Raman spectroscopy. If we map it out, now green is pyrite, red is quartz, and blue is organic material, that kerogen we talked about. In some cases, we find what appear to be films of organic matter on the edges of these pyrite grains. And that could indicate that maybe microbes were colonizing these reduced minerals. And in some cases, this one doesn't show it, but previous work that other people did before we wrote this proposal to start doing this work found evidence potentially of oxidation of the pyrite on the rims. So maybe the microbes were, doing some chemistry along the rims of these and oxidizing them, or just producing oxygen, which then oxidized it. So this is still early days in this project, but here's just some work on, again, not microfossils themselves, but what microbe fossils might have been, or what the microbes might have been doing at the time. Again, this is 3.2 billion years ago. long before the, at least 700, 800 million years before the Great Oxygenation event. Okay, so just some quick conclusions. We've found some interesting fossils, but lots of other people are too. It's not just us. Starting to reveal a diverse microbial biosphere in the Archean. Still not finding nearly as much as we find in much earlier time periods. But more work is still needed. We need to find more fossils. We need to do more detailed morphological analyses, more detailed in situ geochemical analyses, and maybe try to determine some new biosignatures. And there are people doing this too, to help detect evidence of ancient life, because this can help us also look for evidence of life when we bring back samples from Mars. And now, in the last few minutes, I will talk about some of the stuff that's going on Mars. Okay, so this is a busy diagram, and I don't want to talk about every panel on it, but it's a useful one. I found it in this paper by someone named Sazalov. I don't actually know Sazalov. Comparing Mars on the top with Mars' history with Earth's history. So again, time is on the bottom. 0 is today. 4.5 is 4.5 billion years ago, because both planets formed at the same time, they're the same age. If we look at Earth, this what we see here is this surface age sort of also represents what I talked about before with the age of the rocks that exist at our surface. We don't have many rocks left over. There also was the amount of rock that was made kind of ramped up over time. We have Earth's atmosphere changed over time. We've had a hydrosphere, we think, throughout. Mars has been different than that, right? Mars's atmosphere had a thick atmosphere at the beginning, most likely, based on a lot of evidence we see, and then it went away, largely. It's still there somewhat, but it's a weak atmosphere. And because of that, too, the hydrosphere has largely gone away or gone underground. And the surface age of Mars is kind of the inverse of Earth. Most of the rocks on Mars are really old. They're billions of years old. And then you get some that are younger than that. On Earth, it's almost all young rocks because the old ones have been destroyed. But the point is that Mars, when it did have liquid water in lakes and channels, there's not really strong evidence for it having a big global ocean, but I guess there's some. I'm not an expert on that. But there's certainly water there now, and there would have been more water in the past. And when it had more of an atmosphere, is right around the same time as Earth's oldest fossils and before. And Earth's oldest fossils doesn't mean that's when life formed on Earth. That's the earliest evidence for life we have, which means life got started earlier than that. So if life got started on Earth then, it could have gotten started on Mars then at the same time, because it's another rocky planet. It had all the same stuff that Earth did early on. It is further from the sun, has a little less sunlight. It is definitely colder. Sun was dimmer at the time, but somehow Earth had a liquid water on its surface, so Mars could have too. So anyway, that's why we are interested in looking at Mars, and particularly looking at the old rocks on Mars. We're not looking at the younger lava flows on Mars, because what's the point? Nothing can really live at the surface that we can imagine well, there's lots of reasons to look at those rocks on Mars. I'm talking about from the point of view of astrobiology. In terms of planetary history, absolutely. Let's look at young lavas. So the potential biosignatures on Mars, and I'm definitely running out of time, so I'm going to go a little faster. But I'm happy. I want to leave some time for questions too, and I'm happy to go back to anything. But these are the different types of biosignatures that we find on Earth, and we would expect to find, we could find similar things on Earth, on Mars, if life ever got started on Mars. We have no idea if life ever got started there. But we could find organic biomarkers like we talked about before. That's what's represented in the upper left corner by those little squiggly organic chemical diagrams. We could find minerals that were, produced by organisms. We could find stromatolites, or those, microbialite, like things we see here. Actual cells, like I study, that'd be great. But again, difficult to find. Different chemical ratios of different elements, and of course, isotopes, like I mentioned, but we haven't really talked much about. But just about all of these In fact, everything except probably the stromatolites, and even those are essentially going to require, well, currently it was going to require bringing samples back to Earth because it's very difficult to, those fossil bacteria, too small to see with the imagers we have. And also the preparation you need of making those thin sections is difficult. It's hard to do that with a rover. And you can't find it just by looking at the surface of a rock that you've abraded away a little bit. Organics, we could potentially detect organics, but they have to be in higher concentrations. If we find low concentrations, we could detect that with labs on Earth easily. It's harder to do with a rover. Isotopes are hard to measure. It's been done on Mars, but it depends on the isotope system. Anyway, bringing all this stuff back makes life a lot easier. But you are limited in which samples you bring back and how much of each one you bring back. If you go to Mars with your instruments, you've got the whole planet, but you're then limited by the detection limits of your instruments. All right, so the Mars 2020 mission, which I'm part of, one of the main things it's doing is searching for evidence of ancient life. That is one of the overarching goals of this mission, search for evidence of ancient life, both with the rover, but also with samples. So, and in fact, this mission is largely a sample collection mission, which will then, which the rover is there to support. The rover's doing it, but all the instruments on the rover are there to document the rocks and the geologic context for the samples. It's a different philosophy than the Curiosity rover, the other rover that's still currently active on Mars, which has an ability to drill rock and collect sample, but it measures it with the rover, and it's there to support the rest of the instrument, rest of the rover. Here it's the other way around. And so it's very different, which means like we're constantly looking for rocks to sample when some of the people running the instruments are like, oh, I really want to just study all these rocks because I want to know more about them, but we got to keep moving to collect samples. Yeah, so and this is the first leg of Mars sample return. And there's the rover. The first leg meaning we're collecting the samples and they'll be on the surface. NASA and ESA, the European Space Agency, now have to design and implement the next couple of missions to land and collect the samples and bring them back to Earth. This rover has no capability of bringing anything back to Earth. Okay, and so we're here in a place called Jezero Crater, which is not that large impact basin. That's called Isidis. Jezero is a small, about 40-kilometer crater on the edge of this large crater called Isidis. And why are we there? Well, there is a delta deposit. So some people think it might be more of a fan, it might be more alluvial than a true subaqueous delta for the full period of time. There is some evidence for actual delta deposition if you're a sedimentologist, which I'm not really. You know what that means. But there are other portions of it that the sedimentologists say, maybe it's not actually, maybe it's more with water bringing sediment in, but not depositing it underwater. It's being deposited and then the water flows on. Mars has a very different history than Earth, but it's definitely a sedimentary environment. So that's why we're there. Places where there's water are places where, life can do its thing. And, we can find evidence of chemical alteration from that water and from life and deposition of fossils and biomarkers. This is possibly what Jezero Crater looked like early on. This is a modern image that's been painted over basically to show there's an inlet channel in the upper left and an outflow channel on the right there. We see evidence of that, but obviously it doesn't have water in it currently, but that's maybe what it looked like back when it formed. And there's only about 5 minutes left, so I might just sort of skip to the conclusion here at the end. I'm sorry for running over a bit. Instead of me just running through everything and running completely out of time, I will jump to this last bit here. And, just to say, what we found so far are sedimentary rocks, both from, deposited in the lake by a river, by flood deposits, and igneous rocks, both intrusive and extrusive, or at least some of the cases we're trying to still decide which it is. The samples include multiple habitable and potentially habitable environments because they were water deposited. There's mudstone, sandstones, conglomerate. We're going to be doing some more sampling ahead and outside of Jezero, assuming all goes well and funding continues. We could potentially find impact-induced hydrothermal systems, pre-impact crusts, so rocks that are like 4 billion years old and older. We don't have any of those rocks on Earth. So even putting aside astrobiology, we can learn about planetary formation from rocks that we don't have on Earth, right? They just don't exist anymore. And again, returning these samples would allow us to perform these analyses that are not even possible with a rover currently. All right, that's it. Well, maybe I'll jump back to that slide. And if anyone has any questions about the mission or Mars or sample return or something, I do have other slides in this deck I could skip to quickly and show stuff if anyone's interested. But
questions in the room?
Yes.
One would think that some of the imaging techniques that we have to look at some of these things would be, I mean, you showed some pictures. I don't mean you should throw any shade on your pictures or anything, but you know, they are real small, but there are, you know, advances so we can see some of these creatures and rocks a little bit better than. You know.
Yes, there are. I mean, there are people who, yes, people use transmission electron microscopy to look at. You have to prepare your sample very carefully with like a focused ion beam milling, which is a technique to make a very thin wafer of rock. You can look at the ultrastructure of a cell wall, and that's one way to get more evidence that these things were actually biological. So it's not just the gross morphology of the whole thing, but you don't always get that ultrastructure. Ultrastructure meaning I should point out, it's largely done with younger fossils. When I say younger, I mean like maybe 1.5 billion years old, where we had larger, more robust organisms at the time. They may even have been eukaryotes. And the cell walls had more features to them. And we can see that in modern ones, and you can compare that with these fossils and say, oh, we see these details in that cell wall. That gives us more confidence they're biological and maybe that they're eukaryotes. But it doesn't always get preserved. So you don't want to necessarily say we have to find that in order to be convinced that it's alive. People are using micro CT, what was the micro X, is it XCT, X-ray computed tomography? Yeah, like you have over in the Jackson School downstairs. I got to see that today. That was a really cool facility. People have been using that, not so much to look at these microfossils because they're rather small. And from what I understand, the resolution is maybe it'd be good enough to see those things with some detail. But you also need a contrast between the masses of those elements. So the difference between carbon and silicon is not so great if you're looking at organic matter in a chert rock. If you're looking at something that's made of metal inside chert, you're going to see you see a big contrast. You can image those things well. But people have looked at larger objects like stromatolites to look at those, some internal features of those with micro XCT. I also have one of my students I showed you before, Andrea Corpolongo. She's been doing some work imaging some of these stromatolites, but actually serially sectioning them. She has a slab of rock. You can see on the surface of it, the layers. She images it on a flatbed scanner, polishes it off, images it again, polishes it, images. Well, she has an undergrad helping her, a couple of undergrads, and then she can reconstruct a 3D image of that. She's about to publish that. I didn't want to scoop her, pretending it was my work or anything. Yeah.
I could be completely off this. I remember reading something like getting back to this preservation, and assuming that these were living on the surface because they need sunlight, and you lose your magnetic field. Destruction via radiation and not being able to get deep enough with the coring tool.
On Perseverance.
This would be an issue that you could not get deep enough. That even if you had something in there that had been destroyed, by radiation in the interim.
That is a concern. One thing is though too, is it's sometimes difficult to know how long that surface has been exposed. Because the surface it's been sitting there without, you know, water on it for a very long time. But these rocks do get eroded by wind. And sometimes we have found some rocks that have broken off from an outcrop. And so that is a relatively new face. It might be hundreds of millions of years old or something, but it's maybe not 3 billion years, 3.5 billion years that rock has been exposed to solar radiation. So, but it doesn't necessarily take that long for it to. So yes, that is a concern of if you have a rock that's been just being hit by cosmic rays and solar radiation, you know, maybe getting down in about that far, which is about the size of the length and size of the cores are about my pinky. That might not be deep enough to get below that realm of destruction. And so if there was organics in the rock, they just may not be there. But, you know, it's the best we can do currently. The Europeans are sending ESA is sending hopefully soon, or in a few years, their mission, which is going to be able to drill down about 2 meters, but I'm pretty sure it's only going to be drilling into the regolith. I don't think they have a drill that can drill into rock 2 meters. That would be a feat.
Well, everybody, thank you, Andy, again for coming, and appreciate you all attending.
Yeah, thank you. And if anyone had any other questions and can stick around, I'm happy to answer anything. I did see one more hand in the back, and I apologize we didn't get to your question.